you, and good morning. Thank you so much for having me come speak with you today. So here's the standard disclaimer. I won't read it. So the key learning objective really for today is kind of dovetails on what Eric was saying yesterday about, highlighting the importance of considering the end user when we're developing our labeling. A lot of times we're very good at putting in content, but we often don't think about how that's being communicated to the folks that are ultimately using the instrument. And when you're talking about highly technical information, such as clinical pharmacology, it does become a bit of an issue. So clinical pharmacology-related information probably has one of the largest footprints in the label itself. It informs many sections, and I've highlighted the key ones here in yellow, but there potentially could be others as well. And as generally this information tends to be of a highly technical nature, so it's often a bit of a challenge for healthcare providers who may not have had a pharmacokinetics course in 20 years, or maybe haven't had one at all, to really interpret this information and figure out the bottom line is, how do I prescribe this drug to a patient in a safe and an effective manner, which is what the regulations say the labeling is for. So as part of CDER's prescription drug labeling enhancement and improvement initiative, the Office of Clinical Pharmacology also looked at its own sections of labeling and tried to see how could we communicate this technical information in a more effective manner. How good are we doing? Can we do better? And we tried to do this by taking three approaches. One, of course, is reaching out to industry, academia, and saying, give us some suggestions, work with us on how we can improve this type of information. And then also working within our own agency and with other groups within FDA to come up with some ideas for direct labeling enhancements to enhance communication of this information to the end user, the healthcare provider, the prescriber, and then ultimately to develop policies and guidances so what we're thinking is clear to industry developing labeling, as well as healthcare providers using the labeling, and us regulating and working with the labeling as part of our public health mission. So just a quick overview of some key regulations, just to give us a grounding for some of the stuff we're going to talk about. These are just highlights. The regulations are more extensive, so I suggest that you look at them if you want more specific information. But first, the drug interaction section, which is generally section 7 of the prescribing information for healthcare providers. And so really what must be in that section of labeling is a description of clinically significant drug interactions. I stress clinically significant. As part of that description, there should also be some information about what the clinical implication is. Okay, there's a significant interaction, but what does that mean? Well, it means there's going to be substantial bleeding if you use these two drugs together. Or there could be death if you use these two drugs together. Or you could have a drop in blood pressure, a significant drop in blood pressure if you use these two drugs together. What's the pull-through from a twofold change in concentration? Just putting the magnitude of a change doesn't really tell the healthcare provider what that means clinically. How is this going to hurt my patient? Then, of course, once we've identified something as a clinically significant drug interaction, then, what practical instructions can we give to a healthcare provider to either prevent or manage that drug interaction? So that's another important thing that must be in section 7. If we have information regarding the mechanism of the interaction, so the drug is metabolized by CYP3A4, and therefore a CYP3A4 inhibitor is causing this drug interaction, then information about the mechanism should be in there as well. And then lastly, the section 7 also is where we would have lab interference related information. So that's section 7. That's one big piece of clinical pharmacology related information. And then we have the clinical pharmacology section, which is the other column on this slide. That's generally section 12 of the prescription drug labeling, and there we should have a summary of what's known about the mechanism of action for a drug. We should have information regarding the biochemical and physiologic pharmacologic effects of the drug that relate to safety and efficacy, so the pharmacodynamics. We should have information about exposure response. So as the concentration or the dose of a drug changes, how does that impact the efficacy or markers of efficacy or markers of safety of a drug? And, which is also key, is if we don't have that information regarding exposure response, we need to say that we don't have that information. Similar to the mechanism of action, where we also need to say if we don't know the mechanism of action, we need to say it's unknown. So those are some places where it's very hard to take that section out, because there's always, it has to be either information about exposure response or there is no information. So it's kind of always kind of has to be there. Then in section 12.3, which is the PK section, we have a description of, again, clinically significant pharmacokinetic parameters for the drug or its active metabolite. And this would also include additional details about drug interaction studies or studies in specific populations related to pharmacokinetics that ultimately inform other sections, such as section 7 that we just talked about a minute ago. And then lastly, this is one section where you can have in vitro information. What the regulations say is we could have in vitro information that's pertinent to establish the absence of an effect. We could also have in vivo information to show the absence of an effect. So a lot of times, and we've heard this from healthcare providers, sometimes they like to know it was studied and there's no issue, but they don't need to have a lot of details about it. So that's why if you look at our section 12 guidance, we say in these cases, you could just kind of give a list saying drug X, Y, and Z were studied, no clinically significant drug interactions were found with those. And that's where you would put it in section 12 versus section 7, where we have clinically significant drug interactions. There's some rare cases where you could put negative information like that in section 7, but for the most part, we try not to. So when we started looking at our own labeling, we had an advisory committee meeting back in 2013. And we had physicians and pharmacists on this panel. And we asked them about our labeling. And we said, what do you guys think about it? And they had a lot to say about what was wrong with it. So they said, well, the structure is kind of confusing. There's way too much information in there. It's kind of highly technical. Some of the information is wrong because it's old and newer things have happened. You're not really good at conveying the real risk because it's so covered with context that we can't really see what the bottom line is. You're telling us there's a twofold difference, but what does that mean? Is my patient going to die or is my patient going to get a tummy ache? How I work on these things is going to change that to a lot. So the bottom line was there's no real guidance here. And then we turned around and said, well, what do you guys want in labeling? And they said, well, we want it to be very easy to access and navigate. We want you to minimize all this pharmacokinetic jargon. AUC, time-dependent this, linear that. We really don't know what any of that stuff is. So if you could explain it, if you need it, it's one thing, but could you at least explain it? Have a clinically intuitive structure. So it's very clear that this drug is metabolized by this, and then therefore inhibitors would block this. So it's a very intuitive presentation of information so that they can get kind of what we're saying. Provide clear risk management instructions, so they really don't like use with caution, monitor closely, monitor period. One comment that we always get is, okay, it's a warfarin interaction and you're telling us to monitor INR. Well, we're already doing that. It'd be malpractice not to do that. So that's not really helpful. Do you want us to monitor more, or do you want us to do, So that's kind of the thing. It's not really helpful risk. Omit unnecessary information. So don't have so much context. We're not writing a scientific paper. We're writing an instrument to help safe and effective prescribing. So, do we need all this context, or can we just give them the essential information to make a prescribing decision, a safe and effective prescribing decision? And bottom line, like was discussed yesterday, the labeling really must be up to date. So these are things that we heard from them in 2013. 2 months ago, we had another meeting. This is more focused toward drug interactions, where we had specifically physicians, pharmacists, nurse practitioners, PAs, third-party drug information providers, and a couple of representatives from industry. And we kind of talked about drug interaction labeling as well. And we were hoping we'd hear a lot that we've improved a lot. But actually, we heard a lot of the same things about we need to be clear. We really want the bottom line. We want it consistent. We want it, take away the noise, give us very clear recommendations about what's the problem and what do we do about it. That was the take-home message. So we're going to talk now about a few strategies that we can use to help with the clinical pharmacology-related information that, again, tends to be more highly technical. And some of these you could probably apply to other sections, but really the focus of this is more the clinical pharmacology-related information. So one thing is cross-referencing, and that was brought up a couple of times yesterday. So that really helps with reducing redundancy. By using cross-referencing effectively, you can minimize the repetitive nature of the information. You can have less information in a particular section that would hide the new, important, significant information that we really need to communicate in that section. So having the same exact information in contraindications, warnings, drug interactions, and in clinical pharmacology, doesn't help because you have 4 huge paragraphs. So being strategic in where you put the information based on the regulations and guidances, you could really cut down a lot of the noise in labeling and still communicate the important information to healthcare providers. So the example here is just with drug interactions. So Section 7 for drug interactions would be sort of home base. That's kind of where you put the bottom line. That's where the totality of evidence is considered, and ultimately we say, this is a clinically significant drug interaction, this is what you need to do about it. If you want to know more information about, well, gee, why is it clinically significant? Well, you can go to the clinical pharmacology section. There's a reference there. You'll see the study results. You'll see the magnitude of change. If you're wondering, well, what is the real safety concern? Well, you can go to the warning section. You can go there, and they might say, well, in patients that have had these in the pivotal trial, we've had so many people get hospitalized or whatever. You could have more information about that. And if it's even worse, it could be in the contraindications or box warning section. And then if you see in there in Section 7, it says, well, a dose reduction is recommended. Well, what's the exact new regimen? Well, then you go to Section 2, and the exact regimen is there in Section 2. So by doing that, we've reduced the redundancy of having all that information all over the label, which makes it more cluttered. So cross-referencing is important. Next one we talk about is clinically significant information. So a lot of what we're saying today and a lot of what you see in regulations and guidances say, clinically important information or clinically significant information, not trying to imply statistical significance versus clinical significance. But so one thing to consider, so what I wrote on the left side of the slide is just some questions you should ask yourself. I mean, ultimately, what is deemed clinically important or clinically significant is going to be a review issue. And this is going to be different for every drug. So I'm definitely not going to stand up here and say, well, never put this in the label, because I can tell you tomorrow I'll get a drug where we need to put that in there. So, so I wouldn't do that. So really, it's ultimately going to end up being a review decision based on the totality of evidence. But some things to think about as you're developing labeling is, one, is the information essential for the safe and effective prescribing of the drug? Yes or no? 2, if it's not by itself appearing to be essential for the safe and effective prescribing of the drug, is it part of a picture? Is it informing another part of labeling that is clinically significant? So by that, it makes it part of the bigger picture and therefore clinically significant information. So we have in Section 7, this is a significant drug interaction, increased elevated concentrations resulting in increased bleeding. Section 12, the fact that we put that it's a 3-fold change based on the study is clinically significant when you put the whole package together, right? So we're looking at it as a whole package. And then lastly, of course, is can we omit some of the noise? So again, we're not writing a scientific paper, so we're not looking necessarily to write things for completeness sake. We're trying to inform safe prescribing. So we need to take a step back once we've put in all that important information and say, is there anything in here that's really not important to this decision, for the ultimate decision? And then think about that. So on the right side, I just give some things to think about. Now, all of these examples could potentially be clinically significant. It just depends on the circumstances. That's why, again, I say it comes down to being a review decision. But one thing to think about is if we have pharmacokinetic information from healthy volunteers as well as patients, sometimes we'll see them both put in labeling. There's no clear reason why. Now, of course, if there's a difference, a clinically significant difference, and we need that information to know, well, okay, there's a clinically significant difference, so the drug interaction studies that did in healthy volunteers I need to take into the context of it may be different in patients. Yes, that information is important, but by just putting those two values in there and assuming a healthcare provider is going to make that jump, I think is not good. I think if that's our goal, then we would need to somehow link that, explain that that's what the issue is, right? Detailed results from unapproved dosage forms. Many times I'll see a drug that is an oral formulation, and then we'll see pharmacokinetic parameters such as volume of distribution, clearance from intravenous data. And me being a clinical pharmacologist, I know that that information is incorrect because we should be using apparent clearance because it's related to the bioavailability of the drug. And but for the healthcare provider who may not be a clinical pharmacologist, they don't know that that's not the right number for this formulation. So first, we shouldn't be having unapproved formulations in labeling to begin with, for the most part. And also, putting this in there is actually more confusing. So if you're putting it in there to say, well, we need a volume of distribution, this is all we have, actually putting that in, adding that sort of intentional confusion into the labeling is probably not very helpful. So we need to think about that as well. Why are we putting this in there? Plasma versus whole blood distribution of the drug. Again, sometimes it's very important. If you have a drug that actually works inside the cell, you'd like to know how much of it distributes inside the cell. In that case, very important. Putting it in because we did the study and for completeness sake, might as well throw it in there. Probably not as helpful because the healthcare provider is going to try to figure out, and we've heard them say this, is when we put this kind of stuff in there, they try to figure out why we put it in there. And sometimes they overthink the problem. So we have to think about that too. Multiple volumes of distribution. So sometimes they'll model pharmacokinetics, and it'll be something they call a 3-compartment model, which will end up with 3 different volumes of distribution. And for completeness sake, we might put those 3 volumes in there. But really, is that important to the healthcare provider who doesn't really know what they are? If, for example, the central volume of distribution is the one that's really important, or something. So we have to really think about that. Are we putting it in because it's clinically important, and we can tie that to something on how the healthcare provider could use this information, or are we putting it in just for the sake of putting it in because we have it? And the last one is inactive metabolites. We often see a laundry list of these highly complicated names that just clutter up the metabolism section, and we miss the important information about the one active metabolite versus the 10 inactive metabolites, right? So really, we'd really like to focus on active metabolites. And by active, I mean it could be pharmacologically active from an effect standpoint, or it could be insignificant because it causes some kind of risk issue, adverse reaction or something to that effect. So, those kind of stuff obviously we need, but we don't need to have so much information about inactive metabolites that important information gets lost. So those are things that I think we should think about when we talk about clinical significance. So this is an example of something you might see in section 12.3, the drug interaction studies heading. And the example above is a preferred example. So really, it's just giving the key information about the exposure change. The example below is not preferred because it has a lot of extraneous information. It has the doses, it has the population it was used in, it has information regarding the dose reduction, which should be either in section 7 or in section 2. So, and that's not to say this information is never important. So for example, if you were to tell me, well, the dose of ketoconazole they used is half the dose that we would normally use for this kind of study, and actually these results may actually be a lot worse than we anticipated. So yes, absolutely, then that becomes important information. Or if we have the issue with patients versus healthy patients versus, healthy subjects versus patients being significant, it might be important to say that the study was done in healthy volunteers. But again, the assumption that a healthcare provider is going to put that different information in different parts of this section together and come up with that is probably not good. So we would need that pull through. So if you're saying, I'm going to put the dose in there because the ketoconazole dose is too low, then we need to say something like, this may be higher in this may be higher than expected due to the dose or something like that. We would have to have that pull through to tell them, okay, how do I use this information? That's the key. In vitro information. So a lot of times we'll see, like I said, in section 12.3, there's often in vitro information. It's usually, from a drug interaction perspective, it's usually used to show that the absence of a drug interaction. If you look at our guidance, we have very specific recommendations regarding, tables and whatnot about, when a drug when an additional study needs to be done in humans versus additional study does not need to be done in humans, based on in vitro information. But the in vitro information, so the in vitro information can be used to say there's an absence of an effect, but it can't be used to say there is a clinically significant effect. So therefore, it rarely, if ever, should be in Section 7, right? Because Section 7 is for clinically significant drug interactions, and in vitro information by itself can't do that. It's used sometimes to describe certain pharmacokinetic parameters, specifically protein binding is a big one. Metabolic pathways is another. Those are areas where we might see it as well. Another thing to think about when it comes to in vitro information, because a lot of times we'll see it as a data dump, because I was a reviewer before I was in my current position, and a lot of times you'll just be like, well, everything that was studied, we kind of put it in labeling, right? We have these long laundry lists of, it's a substrate of XYZ, it's an inhibitor of this, it's a transporter of that. But when you're putting that information, think about how the context of that information relative to the human data that we have. Because a lot of times the information might be contradictory. And we have in one place it's saying it's an inhibitor in vitro, but then we have a clinical study that says it's not. So again, you're the healthcare provider reading this thing. What am I supposed to do? Sort of a thing. Healthcare providers don't necessarily read our guidances, so they don't know the thresholds for, even though it inhibits, we're not going to do a human study. So, but when we put in the in vitro information that's an inhibitor, but then we don't do a human study, well, what does that mean to someone who doesn't know why we didn't do that study? So the question is, again, it comes back to, is this essential information, or are we putting it there just to be complete? And is it being more helpful, or is it being more confusing to the person who's actually reading this information? So generally, if something is, we have human data, it supersedes the in vitro data. So we try not to have that confusion if we can avoid it. I mean, I think the negative information is very important, because they do like, the healthcare providers do like to know something was studied. But when it comes to the positive stuff, I think we need to just be very careful and, think about, what we're communicating, and is it confusing to the healthcare provider. And the other thing that we thought about is, can this information be presented in a better way? Could we somehow have a standard table or something like that where we can communicate this information more clearly? And we're definitely open to suggestions on doing that. The example on the right there is just something we came up with we try a lot of different things, but that's definitely not shouldn't be taken as any kind of template or anything. But we're open to suggestions on different ways of communicating in vitro. Modeling and simulation. So model-informed drug development is a big topic these days. So a lot of information that we have now, especially for clinical pharmacology, is based on modeling and simulation-related information. And the question is, how do we address that in labeling? So physiologic-based modeling was an area of my expertise back when I was a reviewer. So I could tell you it's very complex information. It's even complex for the modelers. So the amount of information we put in, we need to really think about. Of course, the important bottom line essential information should go in there. The amount of information we put about the actual model itself should probably be relatively brief, if at all. I would say anything that's important would need to go in. So for example, if we do have a patient versus normal volunteer effect, then yeah, we may need to know that we did the simulations based on a virtual population in normal patients. But if that's not an issue, then do we need to put that information in there? The doses. Again, it's similar kind of rules to what we had with the human studies. And then ultimately, do we need to define it? Do we need to say in a physiologic-based model, in a POP-PK model? Really, if the information is deemed adequate to replace a clinical study, then probably we don't need to call it out as such. Because sometimes healthcare providers will look at that and say, oh, it's modeling, I don't know, maybe I won't go with it. But really, if our intention as an agency is that this is replacing a clinical study and this information is acceptable for that, then we really don't need to put that information in there. I put that, I think that the highest number of in a pop PK model I've seen is like 16 in the label. If you're going to do that, if you really want it badly, just put it like at the top. All the PK was done based on pop PK, and that should be sufficient to let folks know. We don't have to keep repeating it. Next, technical language. This is especially important for clinical pharmacology. A lot of what we do is, considered technical to a lot of healthcare providers because they don't have a lot of pharmacokinetics and pharmacodynamics in their curriculums. They're more therapeutics and whatnot. And we have to remember too is, you as industry, us as regulators, so you've seen this drug for 5-10 years, right? We've seen it for at least, 12 months, right? Or 9 to 10 months, right? I'm a pharmacologist, and so we're very comfortable with this jargon. And we often don't pull ourselves out and say, well, does someone who knows nothing about this able to understand what we're talking about? So I think that that's what we need to do. So when we're writing technical information like the clinical pharmacology section, we have to think about, can we say this in a simpler way? If we can't, then can we add additional pull-through to just tell them how to use this information, what we're thinking with this information? Of course, can this be understood by someone who's not a clinical pharmacologist? That's always a big question. And is the interpretation clinically intuitive? So am I assuming that this person is going to go all over this section to have to figure out what I'm trying to get them to do? Is that what I'm doing? And there's some examples on the right-hand side. So and these came from labels that they're just de-identified and changed a little bit, either labels or submissions. So the first is drug X shows time-dependent PK with a 13% decrease in steady-state clearance. So yes, to me, a regulatory officer who's a clinical pharmacologist who has the access to this NDA at my fingertips, I know exactly what they're doing there. My guess is probably this drug is metabolized by a pathway that auto-induces itself, and that takes a certain amount of time. So therefore, the auto-induction takes place out by steady state, and therefore we're seeing a decrease. That's me. But think about now the healthcare provider who's a family practice doctor in a small town trying to treat this patient, reading this information. Well, what does that mean to them? I Googled time-dependent pharmacokinetics. It's not very intuitive. So things to think about. Well, one, is it clinically important information? Should it be there at all? Is this 13% decrease important? Do we need it in the labeling? If it is, then if it's not clinically important, but we think we should still communicate it anyway, then do we need to just say there's a 13% increase, but it's not deemed clinically significant? Or the other side of the coin would be don't include it at all. Or, it is possible that this information is used to inform dosing, right? It could be in the dosing section that will say, use this initial dose, but then increase it by X amount, by this time period. And then there's a cross-reference to this note saying, but again, is this the way to say it? Could we just say, a 13% increase in drug concentration was observed due to drug X metabolism, self-metabolism, or increased metabolism, or something like that. There's another way to say it where it's not as technical, and it's easy to understand what the bottom line is. The next example is decreasing drug X dose from increasing drug X dose from 50 to 150 once a day resulted in a slightly less than dose proportional increase in drug oxide steady state Cmax. So the issue there is the slightly less than dose proportional. So, yes, we all know what dose proportional is. We all know what not dose proportional is. It means nonlinear, right? But when we get into this sort of slightly, so what it means to me as a regulatory officer, as a clinical pharmacologist, well, what it means to me is, there's a beautiful graph that the sponsor will provide for me that shows dose and shows the AUC and Cmaxes. And the line isn't exactly like that. It's a little bit like that. But it's not like that, or it's not like that. But generally, it's probably not an issue. I'm just paraphrasing from this. Now, there definitely could be places where it's an extremely low therapeutic, high therapeutic index drug, and it's this information is important. But again, is this the way to communicate that fact? So if it is an issue and it affects dosing, and there needs to be a cross-reference from the dosing section to this, then yes, we need to explain it in a different way. But really, are we putting the slightly different just to be just for purposes of completeness, or are we saying it because there is an important reason? You could, my question to the reviewers when I come across these kind of things is, what do you want a healthcare provider to do with slightly less than dose proportional? And if they say, well, nothing, then does it need to be there? If they say something, then I'll say, well, then let's just say that. Whatever they tell me, they'll say, well, I want them to realize that this is why we changed the dose in this. Then that we need to communicate it better that way. The last one is one of my favorites. So drug oxide is an inhibitor of BCRP and PGP transporters with IC50 values of 50 to 273. So IC50 values are a way that we look at. It's the inhibitory concentration for 50%. It's a way of looking at how a drug inhibits another drug. Actually, if you look at our guidances, we actually recommend a different parameter, Ki, but for purposes of this example, we'll go with IC50s. And the reason I bring this up is, so this was presented in this label, and the purpose of it is they wanted to show that the drug would inhibit at the concentrations that you can get, when dosing the drug. But the problem was that the concentrations they gave are higher up in this section were in nanograms per ml. So they assumed that the physician would look at this information, know why they put it there, and then go up to the exposure information, mentally convert it from moles to grams to moles, that kind of thing from chemistry, and then put together what the issue is. So really, it's probably better just to say it inhibits, it doesn't inhibit, that kind of thing. This is almost too much information. So, clarity and readability. So you've always heard this, the same old thing, use the active voice. We've heard this from healthcare providers as well. I'll admit I'm not always the best at it. Mongthuong usually has to correct me sometimes because I've been doing labeling too long. But if you work at it, you get into the practice of thinking more active voice. Again, try to only provide sufficient detail to inform the prescribing decisions. Try not to give so much context that the key message that you want to present is lost. And we do see that a lot in that. So have your actions clear and specific. Try to reduce redundancy, use brevity. Definitely, we've heard this from healthcare providers, try to avoid the closely monitor, the use with caution, these sort of non-value added things. Use white space text attributes, bolding, bullets, whatever, to help break up complex or extensive information. Wherever possible, use tables or figures to help enhance readability. Now, there's no preference. So in our Section 12 guidance, we don't say, well, you must use a table for this or you must use a figure for that. So it's really based on the breadth and complexity of the information that's presented, whether maybe a table would work better or just text would work better or a figure or a graph or something would work better. It's kind of hard to pigeonhole that information, so that's sort of more of a case-by-case kind of thing. But the important take-home message is all those alternatives are open to try to make our labeling better, and we hope that industry uses them. So we're just going to look through some examples now. And again, none of these are meant to be templates or requirements or anything. They're really meant to say we're open to a lot of different suggestions, and we really need industry to work with us as well as academia to work with us to try to make these things better. So we have lots of different ways of doing things, just to show you that we're open to a lot of different ways of doing things. So this would be an example of a Section 7 labeling that you probably saw prior to 2015. So there's not much white space. There's not a lot of breaking up the information. It's just a glob of information. This is a newer way that we've been working to do it. We try to add more white space. We try to break up information. We try to structure it in more of an intuitive way. Another option would be to use a table. And when we presented this to the healthcare providers, they actually did like this, more compartmentalized, because it really showed them where all the information was. And whether or not to use examples, by the way, there's no regulation that requires examples. So it's really up to industry. Just keep in mind that a lot of times we try, it's hard for healthcare providers because the examples are inconsistent. They often aren't comprehensive. So we need to think about whether or not the benefit versus positive negatives of including examples. What we've heard from the third-party information providers and what we've seen based on the questions they send us is that they do know, where those inhibitors are. So so if we assume that healthcare providers are using a lot of these more third-party references that are informed by labeling, they would get those examples that way. So just something to think about. This is another example of a table you might see in Section 7. This is for when you have an extensive number of drug interactions. So like the antiviral drugs, where they have like 50 drug interactions, and you can't even put the structured table I showed you before in there because it would take up the entire label. This is a way of maybe sort of giving the minimum of information. But again, this is just an example. We're open to more suggestions of how to describe extensive drug interaction information in Section 7. So now moving to the clinical pharmacology section, Section 12. So again, it depends on the type of data you have. So if you have a biologic with very limited PK information, text is fine. If you have a drug where it has not only the drug but multiple active metabolites that you have to account for the PK of, it becomes very complicated. And in those cases, you may want to consider a table like this here. So this is more of a structured kind of a table. Again, not a template, but just an idea. But ultimately, we would love to get ideas from industry and be able to get to a point where we could come up with sort of a more standardized structured table for different scenarios. Another good place for tables for PK information is when you have a fixed combination product. So in the past, you'd have to have these huge paragraphs for each component of the product. And now you could use a table and have a column B for each drug component of the product. And this actually worked out really well for decluttering the label. Now this is something that our fellow developed, Yang Ho, and she's a pharmacist who's also a medical illustrator, and we challenged her with, can we take some of the more common pharmacokinetic parameter information and describe it as a figure? And maybe in giving it as more of a standardized figure, both explain what we're talking about to people who aren't clinical pharmacologists and provide the information. And this is what you came up with, which I thought was kind of cool. Again, not a requirement, not something that we're telling industry to do, but something that we could say that, this is an option too. I mean, think about it. This could be something that we could do for more common information. So just letting you know that we're open to all different kinds of ideas. This is an alternative display that would be in Section 12 to describe the drug interaction studies. So the more details that would inform Section 7. So this has all the key information from the studies, and rather than having each study listed separately, listed in one place. This is the same information, but as a graphic presentation. There's no preference whether we use one or the other. One question I often have is how useful are 90% confidence intervals to healthcare providers? When we asked them that question, it didn't seem like they were all that useful. They were more interested in like a range. They want to know like what's the best and worst case scenario type thing. That's more informative to them. So again, it comes down to, what I need as a regulator and what you need as industry to give to a regulator to make a regulatory decision is often very different than what a healthcare provider needs to make a prescribing decision. And 90% confidence intervals are essential for regulators to make a decision about is there a difference. But as far as in practice, where healthcare providers are more used to 95% confidence intervals, it looks kind of like a range, it could actually not be as helpful. I put this example in here to highlight another important issue. So this actually came out of a label. If you look at this figure here, you would think that the items in the red box are clinically significant drug interactions, but actually none of these are. And the reason I put this there is to highlight the point that if you use this type of presentation, it's very important for you to delineate what is clinically significant and what is not, because even with 90% confidence intervals, because there's a difference in 90% confidence intervals, there may not be a clinically significant difference from point of risk or efficacy and that kind of thing. So that's a very important thing to think about as well, is to make it clear to the healthcare provider when we're giving all these details, which of these details are clinically significant. And then this is just a quick example from dosage and administration section. If you have a complex dosing regimen, which this is, it's based on how long the potential co-drug is used and what dose it's used. It's very difficult to do in text, which is on the left. It might be easier to do sort of as a flow chart kind of a thing on the right. Again, a different way of thinking about things. And of course, you know, if we don't put all this context in, where could healthcare providers get some of that information? Well, we do have our reviews, at least for the NMEs, posted on drugs at FDA. Of course, they can always contact the company and ask for additional information. But there are ways to get additional context for academics or people who would need that information. So, thank you. Our challenge interaction, section 12.3 is text, table, figure, A and B, no preferred format. How many say, well, shout it out. E, great. Perfect, thank you. And we're ready for the panel, I guess.
Go to the next slide, if you would, Joe.
Oh, sure. Yeah.
All right, and we're going to go ahead and do a very short panel. We've got about 4 or 5 minutes to hit some questions. As announced earlier, Mongthuong is joining Joe for the panel. If there's any questions in the room, please come on up to the microphone. Otherwise, let's go ahead and take our first online question, please.
For a drug-drug interaction and the sponsor has single-dose PK data and has steady-state PBPK modeling data, does FDA put more weight or importance on the human single-dose data even if the steady-state modeling data is more clinically meaningful for a chronic administration drug?
That's really a review issue. It would depend on, you know, having been a modeling person myself, it would depend on, is the model itself acceptable? Is it acceptable to you know, replace a clinical study? So really that's going to be something that's going to be more of a case-by-case kind of a thing. But if it's deemed acceptable and it's in labeling, you know, as far as a labeling perspective, you know, it would depend on ultimately, like I say, it's a review issue. So if they were to assume that they were of equal weight, then it should be presented in the labeling as them being, you know, equal. As far as the decision, you know, modeling stronger than human data, that itself is a review issue. And, you know, we have a group that just reviews PBPK models and works with that. So that would be a question more for our pharmacometrics division, how they would handle that. But again, if it was deemed acceptable, then in labeling would probably be handled the same way as the human data.
Okay, our next online question, please.
For older drugs that are very strong or potent cytochrome P450-3A inhibitors that are well known to cause clinically significant interactions, what is the expectation from the agency to continuously add new drugs that are approved each year that would potentially interact with strong cytochrome P450-3A inhibitors if the USPI already has a broad statement?
I'm assuming this question relates to including examples in Section 7. And if that's the case, again, as I mentioned in my talk, there's no regulatory requirement to put examples into labeling. Obviously, as Eric stated yesterday, and as I said in my talk, you know, that labeling should be up to date, but it would probably be impractical to have every single CYP3A4 inhibitor known to man in a label. Again, the decision from both the industry and regulatory part is including a limited list that may be inconsistent with other drugs in that class even. Is that helpful or is that not helpful? So I think that's the bigger question with regard to the example issue. As far as updating it, obviously Cross-labeling is a big issue. I think it would depend on risk. I think, if the drug interaction that we're talking about with this new drug has some very serious implications, then should it be included in the example list? I would think maybe, probably, but again, that would ultimately be a review decision with the division.
This one. I would just add that we need to approach labeling as if there is a list, that is not a comprehensive list, both from a labeling side, from a reader side. So it's not intended to be all-inclusive. So that should not be the assumption when a physician is trying to read and interpret labeling.
Okay, and I think we can squeeze in one more online question before we go to break.
When there might be a drug-drug interaction of interest to physicians, but the data shows that there is no clinically significant effect, should this information be conveyed to physicians in the label or leave it out because it is not clinically significant?
So as Joe mentioned in his presentation, Section 7 is generally reserved for clinically significant interactions. We do think that information is important if it was studied and deemed not to be clinically significant. Typically, that information would be described or communicated in Section 12.3 under the drug interactions studies subheading. So it would still be in perhaps not in Section 7, because again, that is reserved for clinically significant interactions.
All right, and that is all the time that we have left for questions. Please help me thank Joe and Mongthuong for sharing their